Ribozymes are RNA molecules that have the ability to catalyze specific biochemical reactions, including RNA splicing in gene expression, similar to the action of protein enzymes. Researchers who are investigating the origins of life through the RNA world hypothesis have been working on discovering a ribozyme with the capacity to self-replicate, which would require it to have the ability to catalytically synthesize polymers of RNA. This should be able to happen in prebiotically plausible conditions with high rates of copying accuracy to prevent degradation of information, but also allowing for the occurrence of occasional errors during the copying process to allow for Darwinian evolution to proceed. A protocell is a self-organized, endogenously ordered, spherical collection of lipids proposed as a stepping stone toward the origin of life. A central question in evolution is how simple protocells first arose and how they could differ in reproductive output, thus enabling the accumulation of novel biological emergences over time, biological evolution. Although a functional protocell has not yet been achieved in a laboratory setting, the goal to understand the process appears well within reach. Compartmentalization was important in the origins of life. Membranes form enclosed compartments that are separate from the external environment, thus providing the cell with functionally specialized aqueous spaces. As the lipid bilayer of membranes is impermeable to most hydrophilic molecules, cells have membrane transport systems that achieve the import of nutritive molecules as well as the export of waste. It is very challenging to construct protocells from molecular assemblies. An important step in this challenge is the achievement of vesicle dynamics that are relevant to cellular functions, such as membrane trafficking and self-reproduction, using amphiphilic molecules. On the primitive Earth, numerous chemical reactions of organic compounds produced the ingredients of life. A prokaryote is a single-celled organism that lacks a nucleus and other membrane-bound organelles. Besides the absence of a nucleus, prokaryotes also lack mitochondria, or most of the other membrane-bound organelles that characterize the eukaryotic cell. It was once thought that prokaryotic cellular components within the cytoplasm were unenclosed, except for an outer cell membrane, but bacterial microcompartments, which are thought to be simple organelles enclosed in protein shells, have been discovered, along with other prokaryotic organelles. Molecular studies have provided insight into the evolution and interrelationships of the three domains of life. The division between prokaryotes and eukaryotes reflects the existence of two very different levels of cellular organization. Only eukaryotic cells have an enveloped nucleus that contains its chromosomal DNA, and other characteristic membrane-bound organelles including mitochondria. Distinctive types of prokaryotes include extremophiles and methanogens. These are common in some extreme environments. The eukaryotes are usually now regarded as having emerged in the archaea or as a sister of the Asgard archaea. This implies that there are only two domains of life, bacteria and archaea, with eukaryotes incorporated among archaea. Eukaryotes represent a small minority of the number of organisms, but, due to their generally much larger size, their collective global biomass is estimated to be about equal to that of prokaryotes. Eukaryotes emerged during the Proterozoic Eon. Eukaryotic cells typically contain other membrane-bound organelles such as mitochondria and Golgi apparatus. Chloroplasts can be found in plants and algae. Prokaryotic cells may contain primitive organelles. Eukaryotes may be either unicellular or multicellular, and include many cell types forming different kinds of tissue. In comparison, prokaryotes are typically unicellular. Animals, plants, and fungi are the most familiar eukaryotes. Other eukaryotes are sometimes called protists. Proterospongia is a genus of single-celled aquatic organisms which form colonies. It is of interest to scientists studying the mechanisms of intercellular signaling and adhesion present before animals appeared. Basically, this is how marine sponges are made, since sponges are including in the animal reign. At this stage we divers from plants, but not from mushrooms yet. A common characteristic of opisthecons is that flagellate cells, such as the sperm of most animals and the spores of the chytrid fungi, propel themselves with a single posterior flagellum. It is this feature that gives the group its name. In contrast, flagellate cells in other eukaryote groups propel themselves with one or more anterior flagella. However, in some opisthecont groups, including most of the fungi, flagellate cells have been lost. Their characteristics include synthesis of extracellular chitin in exoskeleton, cyst spore wall, or cell wall of filamentous growth and hyphae, the extracellular digestion of substrates with osmotrophic absorption of nutrients, and other cell biosynthetic and metabolic pathways. Finally, we begin to see true animals here. Some phylogenists once speculated the sponges and eumetozoans evolved separately from different single-celled organisms, which would have meant that the animal kingdom does not form a clade. However, genetic studies and some morphological characteristics, like the common presence of choanocytes, now unanimously support a common origin. It has been suggested that one type of molecular clock and one approach to interpretation of the fossil record both place the evolutionary origins of eumetozoa in the Ediacaran. However, the earliest eumetozoans may not have left a clear impact on the fossil record, and other interpretations of molecular clocks suggest the possibility of an earlier origin. Bilateria are animals with bilateral symmetry as an embryo. This also means they have a head and a tail, as well as a belly and a back. Nearly all are bilaterally symmetrical as adults as well. The most notable exception is the echinoderms, which achieve secondary pentaradial symmetry as adults, but are bilaterally symmetrical during embryonic development. Having a front end means that this part of the body encounters stimuli, such as food, favoring cephalization, the development of a head with sense organs and a mouth. The body stretches back from the head, and many bilaterians have a combination of circular muscles that constrict the body, making it longer, and an opposing set of longitudinal muscles that shorten the body. These enable soft-bodied animals with a hydrostatic skeleton to move by peristalsis. Nephrozoa is a major clade of bilaterians, divided into the protostomes and the deuterostomes, containing almost all animal phyla and over 1 million extant species. These animals have Hox genes, a sequence of genes aligned along the body's length. Each is expressed in a body segment and activities the genes corresponding to its structures. Mutations in the Hox genes alter the large-scale body architecture without having to change each single eye. The muscles are longitudinal and circular, so they can change the body proportions while overall volume is conserved by inner fluid. Also, these organisms have an obligate sexual reproduction with separate sexes. Food enters through mouth in the front of the organism, and waste leaves through anus in the rear. Deuterostomia are animals typically characterized by their anus forming before their mouth during embryonic development. The group's sister clade is Protostomia, animals whose digestive tract development is more varied. Except for the echinoderms, both the hemichordates and the chordates have a thickening of the aorta, homologous to the chordate heart, which contracts to pump blood. This suggests a presence in the deuterostome ancestor of the three groups, with the echinoderms having secondarily lost it. These organisms have rudimentary eyes called pinhole eye. Each receptor receives only light from a very specific direction, forming an image on the retina. A segmented heart pumps the blood and water leaves through pharyngeal slits that may contain gills. All chordates possess, at some point during their larval or adult stages, 5 synapomorphies, or primary physical characteristics, that distinguish them from all the other taxa. These 5 synapomorphies include a notochord, dorsal hollow nerve cord, endostyle or thyroid, pharyngeal slits, and a postanal tail. The name comes from the first of these synapomorphies, the notochord, which plays a significant role in chordate structure and movement. Chordates are also bilaterally symmetric, have a coelom, possess a circulatory system, and exhibit metameric segmentation. In addition to the morphological characteristics used to define chordates, analysis of genome sequences has identified 2 conserved signature indels in their proteins, cyclophilin-like protein and mitochondrial inner membrane protease, which are exclusively shared by all vertebrates, tunicates and cephalochordates. Also, they have dorsal hollow spinal cord that contains most neurons of the body and widens into a brain in the front. All vertebrates are built along the basic chordate body plan, a stiff rod running through the length of the animal, with a hollow tube of nervous tissue above it and the gastrointestinal tract below. However, the notochord is segmented by vertebrates made of bone or cartilage. There is a possible factor of the Cambrian explosion, more oxygen, more carbonate and phosphate for skeletons, supercontinent breakup, depletion of thick bacterial mats, arms race with predators, multiplication of Hox genes. The blood begins to turn red while hemoglobin carries oxygen and kidneys appear, this organ composed of nephridia filter blood and expel its waste as urine. A more active lifestyle requires more food, capturing it with cilia and mucus insufficient. A muscular pharyngeal pump draws water into mouth and out through hills. These animals have camera eyes with a cornea, the lens focuses light on the retina, forming a distinct image. Ostracoderms are the armored jawless fish of the Paleozoic era. The term does not often appear in classifications today because it is paraphyletic and thus does not correspond to one evolutionary lineage. An innovation of ostracoderms was the use of gills not for feeding, but exclusively for respiration. Earlier chordates with gill precursors used them for both respiration and feeding. Ostracoderms had separate pharyngeal gill pouches along the side of the head, which were permanently open with no protective operculum. They are among the earliest creatures with bony heads. The microscopic layers of that shield appear to evolutionary biologists, like they are composed of little tooth-like structures. These animals have pineal eyes, exposed part of pineal gland that perceives daily and seasonal changes of light. Muscles contract to squeeze water through the gills. As they relax, elastic cartilage bars open the mouth to draw in more. Also, some fish have paired flaps that help the body to keep its balance. The tail fin is often asymmetric. Gnathostomata are the jawed vertebrates. Gnathostome diversity comprises roughly 60,000 species, which accounts for 99% of all living vertebrates. Jaw development in vertebrates is likely a product of the supporting gill arches. This development would help push water into the mouth by the movement of the jaw, so that it would pass over the gills for gas exchange. The repetitive use of the newly formed jaw bones would eventually lead to the ability to bite in some gnathostomes. Newer research suggests that a branch of placoderms was most likely the ancestor of present-day gnathostomes. A 419-million-year-old fossil of a placoderm named Entolognathes had a bony skeleton and anatomical details associated with cartilaginous and bony fish, demonstrating that the absence of a bony skeleton in chondrichthyes is a derived trait. Early lobe-finned fishes are bony fish with fleshy, lobed, paired fins, which are joined to the body by a single bone. The fins of lobe-finned fishes differ from those of all other fish in that each is borne on a fleshy, lobe-like, scaly stalk extending from the body. The scales of sarcopterygians are true scaloids, consisting of lamellar bone surrounded by layers of vascular bone, dentine-like cosmine, and external keratin. The morphology of tetrapodomorphs, fish that are similar looking to tetrapods, give indications of the transition from water to terrestrial life. Pectoral and pelvic fins have articulations resembling those of tetrapod limbs. The first tetrapod land vertebrates, basal amphibian organisms, possessed legs derived from these fins. Many early sarcopterygians have a symmetrical tail. All sarcopterygians possess teeth covered with true enamel. Lungs form a vascularized pouches of the gut. Life in oxygen-poor freshwater forces the fish to obtain it by swallowing air. Marine fish drink seawater and expel excess salt through gills. Freshwater fish absorb salt through gills and expel excess water in urine. Among the characteristics defining tetrapodomorphs are modifications to the fins, notably a humerus with convex head articulating with the glenoid fossa. Another key trait is the internal nostril. Most fish have two pairs of nostrils, one on either side of the head for incoming water and another pair for outgoing water. Early tetrapodomorphs such as Kennichthyes had excurrent nostrils that had migrated to the edge of the mouth. The notochord remains continuous, but is wholly surrounded by vertebrae along with spinal cord. The fins of the modern coelacanth move in a pattern similar to a walking amphibian. These fish probably lived in murky rivers and spent much time just below surface, with eyes, nostrils and spiracles exposed. Mass extinction in oceans, maybe because of mass bacterial blooms caused by nutrient excess caused by erosion caused by plant growth on land. The bacterial blooms depletes oxygen in water. Many aquatic animals adapt to breathe air. A rise in global oxygen content allowed for the evolution of large, predatory fish that were able to exploit the shallow tidal areas and swamplands as top predators. Several groups evolved to fill these niches. The most successful were the Ilpisteostygalians. In such environments, they would have been challenged by periodic oxygen deficiency. Being shallow water fishes, the Ilpisteostygalians evolved many of the basic adaptations that later allowed the tetrapods to become terrestrial animals. The most important ones were the shift of main propulsion apparatus from the tail fin to the pectoral and pelvic fins, and a shift to reliance on lungs rather than gills as the main means of obtaining oxygen. Both of these appear to be a direct result of moving to an inland freshwater mode of living. The specific aquatic ancestors of the tetrapods and the process by which they colonized Earth's land after emerging from water remains unclear. The transition from a body plan for gill-based aquatic respiration and tail-propelled locomotion to one that enables the animal to survive out of water and move around on land is one of the most profound evolutionary changes known. Tetrapods have numerous anatomical and physiological features that are distinct from their aquatic fish ancestors. These include distinct head and neck structures for feeding and movements, appendicular skeletons for weight-bearing and locomotion, more versatile eyes for seeing, middle ears for hearing, and more efficient heart and lungs for oxygen circulation and exchange outside water. Plant growth outpaces decomposition, removing carbon dioxide from air and locking it into coal beds, causing an ice age as the greenhouse effect weakens. Able to move on dry land, these amphibians remain dependent from freshwater for skin hydration and reproduction. The groups traditionally assigned to Reptiliomorpha differed from their contemporaries, the non-Reptiliomorph temnospondyls, in having a deeper and taller skull, but retained the primitive kinesis between the skull roof and the cheek. The general build was heavy in all forms, though otherwise very similar to that of early reptiles. The skin, at least in the more advanced forms, probably had a watertight epidermal horny overlay, similar to the one seen in today's reptiles, though they lacked horny claws. Their terrestrial lifestyle combined with the need to return to the water to lay eggs hatching to larvae led to a drive to abandon the larval stage and aquatic eggs. A possible reason may have been competition for breeding ponds, to exploit drier environments with less access to open water, or to avoid predation on tadpoles by fish, a problem still plaguing modern amphibians. Whatever the reason, the drive led to internal fertilization and direct development. Land looks like dense swamps and jungles of tree ferns and club mosses inhabited by giant scorpions and millipedes. Cooler and drier conditions favor small tetrapods able to grow quickly and seek shelter in burrows. Reptiliomorph have small scales of keratin, a wear-resistant, waterproof fibrous protein with strands bound by sulfur bridges. The first amniotes, referred to as basal amniotes, resembled small lizards and evolved from the amphibious reptiliomorphs about 312 million years ago, in the Carboniferous geologic period. Amniotes spread around Earth's land and became the dominant land vertebrates. They soon diverged into two clades, the synapsids and sauropsids, whose lineages both still persist today. All three main features listed above, namely the presence of an amniotic buffer, water-impermeable cuties and a robust respiratory system, are very important for amniotes to live on land as true terrestrial animals. The ability to reproduce in locations away from water bodies, better homeostasis in drier environments, and more efficient air respiration to power terrestrial locomotions, although they might still require regular access to drinking water for rehydration like the semi-aquatic amphibians do. Because the amnion and the fluid it secretes shields the embryo from environmental fluctuations, amniotes can reproduce on dry land by either laying shelled eggs or nurturing fertilized eggs within the mother. Amniotic egg has fibrous shell for support and gas diffusion. Larger eggs with large, non-cellular yolk, retains fluids, can be laid in dry soil or in open air. Claws and scales are made in keratin. The pelycosaurs appear to have been a group of synapsids that have direct ancestral links with the mammals, having differentiated teeth and a developing hard palate. The pelycosaurs appeared during the late Carboniferous and reached their apex in the early part of the Permian, remaining the dominant land animals for some 40 million years. Unlike lepidosaurian reptiles, pelycosaurs lacked reptilian epidermal scales. Fossil evidence from some varanipids shows that parts of the skin were covered in rows of osteoderms, presumably overlain by horny scuts. The belly was covered in rectangular scuts, looking like those present in crocodiles. Parts of the skin not covered in scuts might have had naked, glandular skin like that found in some mammals. In the interior of Pangaea, the drier climate favors reptiles over amphibians. These animals have longer and stronger hind limbs, more mobile as they provide the main force for locomotion. Diaphragm separates thoracic and abdominal cavity. Its action lowers pressure in former drawing air into lungs. Temporal fenestra redirects bite force from joint between multiple bones along its edges. Therapsida is a major group of eupelycosaurian synapsids that includes mammals, their ancestors and relatives. Many of the traits today seen as unique to mammals had their origin within early therapsids, including limbs that were oriented more underneath the body, as opposed to the sprawling posture of many reptiles and salamanders. Lumbar vertebrae have reduced ribs for little lateral flexibility. The physiology of therapsids is poorly understood. Most Permian therapsids had a pineal foramen, indicating that they had a parietal eye like many modern reptiles and amphibians. The parietal eye serves an important role in thermoregulation and the circadian rhythm of ectotherms, but is absent in modern mammals, which are endothermic. Evidence from oxygen isotopes, which are correlated with body temperature, suggests that most Permian therapsids were ectotherms and that endothermy evolved convergently in dicynodonts and cynodonts near the end of the Permian. Metabolic adaptation for a constantly high body temperature by living as a large-bodied predator, smaller descendants will have to maintain it with endothermy, else adaptation for nocturnal activity or for fewer, larger offspring. The teeth begin to have differentiation, with large canine fangs. Early cynodonts have many of the skeletal characteristics of mammals. The teeth were fully differentiated and the brain case bulged at the back of the head. Outside of some crown group mammals, all cynodonts probably laid eggs. The temporal fenestrae were much larger than those of their ancestors, and the widening of the zygomatic arch in a more mammal-like skull would have allowed for more robust jaw musculature. They also have the secondary palate that other primitive therapsids lacked, except the therocephalians, who were the closest relatives of cynodonts. The cynodonts probably had some form of warm-blooded metabolism. This has led to many reconstructions of cynodonts as having fur. Being endothermic, they may have needed it for thermoregulation, but fossil evidence of their fur has been elusive. Maybe endothermy appears independently in archosaurs. Crocodiles might have lost it due to aquatic lifestyle. Possible production of milk-like fluid, rich in sugar and lipids, calcium and phosphorus for bone growth, to protect eggs and support young in smaller clutches. Also foramina in the snout bones suggest vibrissae for sensory whiskers. Early mammaliaforms were generally shrew-like in appearance and size, and most of their distinguishing characteristics were internal. In particular, the structure of the mammaliaform jaw and the arrangement of teeth are nearly unique. Instead of having many teeth that are frequently replaced, mammals have one set of baby teeth and later one set of adult teeth that fit together precisely. This is thought to aid in the grinding of food to make it quicker to digest. Endothermic animals require more calories than those that are ectothermic, so speeding up the pace of digestion is a necessity. Like monotremes today, the legs of early mammaliaforms were somewhat sprawling, giving a rather reptilian type of gait. However, there was a general tendency to have more erect forelimbs, forms like Eutriconodonts even having a fundamentally modern forelimb anatomy while the hindlimbs remained primitive. This tendency is in some effect still seen in modern Lemurian mammals, which often have more sprawling hindlimbs. Being nocturnal, early mammals developed their scent at detriment of sight, reduction of color sight to green and blue. As the regulation of internal environment is harder at smaller size, young mammals depend on parental care. The milk is released through skin along milk lines. Panthetheria is an abandoned taxon of Mesozoic mammals. This group is now considered an informal wastebasket taxon and has been replaced by Dryolestida as well as other groups. It is sometimes treated as an infraclass and older books refer to it as being related to Symmetrodonts. These animals have double layer of fur, longer guard hairs and shorter denser underfur. Mammaries with nipples appear along the milk lines. The newborns are like those made by marsupials. They developed arms to crawl to the pouch where the young complete their development. Sex is determined by chromosomes, male, before that it was determined by external temperature. Eutherians are distinguished from non-Eutherians by various phenotypic traits of the feet, ankles, jaws, and teeth. All extant Eutherians lack epipubic bones, which are present in all other living mammals. This allows for expansion of the abdomen during pregnancy. When chewing, the epiglottis separates oral cavity from airflow. When swallowing, it closes trachea to protect it from food. The forebrain develops over the midbrain as olfaction overtakes sight. Corion and Allantois form a syncytial placenta through which the young are fed during their development in the uterus. Plesiadapiforms first appear in the fossil record between 65 and 55 million years ago, just after the Cretaceous mass extinction, although many were extinct by the beginning of the Eocene. They may have been the first mammals to have fingernails in place of claws. The panprimates together with the Dermoptera form the Primatomorpha. Purgatorius may not be a primate as an extinct sister to the rest of the Dermoptera or a separate, more basal stem panprimate branch. Even with Purgatorius removed, the crown primates may even have emerged in this group. These animals adapt to tree-dwelling life in the thick Paleocene forests. They have robust legs to leap among branches and long tail to keep balance. The ears grow larger to focus the sound. Being an evolutionary grade rather than a clade, the prosimians are united by being primates with traits otherwise found in non-primate mammals. Their diets typically are less dominated by fruit than those of the simians, and many are active arboreal predators, hunting for insects and other small animals in the trees. Related to their frequently nocturnal lifestyle, prosimians lack the color vision of higher primates. Like most placental mammals, they are in effect red, green color blind. This allows for more rod cells in the retina, which may enhance vision under low light conditions. However, some of them became more diurnal, them have the ability to see three colors, adding back the red, probably to distinguish ripe fruit from unripe. Warm current through the tethys keeps temperature uniformly high, worldwide tropical climate with jungles up to the polar circles. Also, prosimian have forward-facing eyes. By seeing the same objects from slightly different angles, they allow to gauge distance. While primates are often thought of as fairly intelligent animals, the prosimians are not very large-brained compared to other placental mammals. Their brain cases are markedly smaller than those of simians of comparable sizes. However, they have long-term social groups and vocal communication. Parapithecidae is an extinct family of primates which lived in the Eocene and Oligocene periods in Egypt. Eocene fossils from Myanmar are sometimes included in the family in addition. They showed certain similarities in dentition to Condylarthra, but had short faces and jaws shaped like those of tarsiers. These animals have flat finger and toenails, loss of grooming claw, possibly allow more delicate grasp and better tactile sensitivity. They were probably diurnal, mostly herbivorous but eating some insects. The upper lip is fused and the females have a single pair of mammary. The endometrium thickens during ovulation to allow embryo implantation. If no fertilization occurs, it's broken up and shed with menstruations. Propleopithecoids have a number of features in common with extant catarrhines, but also a number of features that are primitive and not found in later catarrhine families. The animals survived the mass extinction of prosimians and other primitive mammals around 34 million years, closure of tethys and the rise of Himalaya block the heat distribution, Antarctica starts to freeze. Scientist models show for population growth of monkeys that evolving species maximize their reproductive rate or their resource-using efficiency. Primates have slow reproduction but extensive offspring care, long lifespan and use tools. Except for gorillas and humans, hominoids are agile climbers of trees. Apes eat a variety of plant and animal foods, with the majority of food being plant foods, which can include fruits, leaves, roots and seeds. Human diets are sometimes substantially different from that of other hominoids due in part to the development of technology and a wide range of habitation. Humans are by far the most numerous of the hominoid species, in fact outnumbering all other primates by a factor of several thousand to one. At this period, grasslands replace rainforests, except near equator, but these animals are still tree-dwelling. The forest offers protection and food. At sites far distant from East Africa, the presence generalized non-monkey primates, of Middle Miocene age, Otovipithecus from cave deposits in Namibia, and Pyrolapithecus and Dryopithecus from France, Spain and Austria, is evidence of a wide diversity of ancestral ape forms across Africa and the Mediterranean basin during the relatively warm and equable climatic regimes of the early and Middle Miocene. The most recent of these far-flung Miocene apes is Oreopithecus, from the fossil-rich coal beds in northern Italy and dated to 9 million years ago. These animals are all omnivorous. We once believed at this stage they were ground-walking bipeds, but the broad pelvis and the developed legs were probably used to walk on branches. They use tools like twigs to catch ants, chew leaves to carry water to stones to crack nuts. These are the last ancestor in this video we share with other living species like chimpanzee, orangutan and gorilla. Orangutan is probably the closest in lifestyle to this stage among all living apes, mainly fruit and leaf eater. The evolution of bipedalism encouraged multiple changes among hominins, especially when it came to bipedalism in humans, as they were now able to do many other things as they began to walk with their feet. Bipedalism was a result of sexual dimorphism in efforts to help with the collecting of food and to help females to be able to care for their offspring better, as they can now use their hands to hold them. Males were the providers, leaving their responsibilities to be protectors and providers of food and shelter. On the other hand, females were the caretakers and had the responsibility to nourish, groom and care for their children. Also, bipedal posture exposes body more to wind and less to direct sunlight and to ground heat. This was a huge adaptation, as it encouraged many evolutionary changes within humans, including the ability to use their hand to make tools and gather food, as well as a larger brain development due to their change in diet. The jaws reduce and let more place for the brain to grow bigger. It was long believed that human ancestors evolved in the drier savanna east of the Rift Valley, while chimpanzees and gorillas stayed in the rainforest, but Sahelanthropus was found in the forest region. The postcranial remains of Australopiths show they were adapted to bipedal locomotion, but did not walk identically to humans. They have a high brachial index when compared to other hominins, and they exhibit greater sexual dimorphism than members of Homo or Pan, but less so than gorilla or Pongo. It is thought that they averaged heights of 120 to 150 centimeters and weighed between 30 and 55 kilograms. The post-canines were relatively large, and had more enamel compared to contemporary apes and humans, whereas the incisors and canines were relatively small, and there was little difference between the males and females' canines compared to modern apes. Most scientists maintain that the genus Homo emerged in Africa within the Australopiths around 2 million years ago. However, there is no consensus on within which species. The hands are more able to hold tools with longer thumb and shorter palm, but not yet able to produce them. Homo habilis, the handy man, is an extinct species of archaic human from the early Pleistocene of East and South Africa about 2.31 million years ago to 1.65 million years ago. Upon species description in 1964, habilis was highly contested, with many researchers recommending it be synonymized with Australopithecus africanus, the only other early hominin known at the time. Habilis manufactured the Oldowan stone tool industry and mainly used tools in butchering. Early Homo, compared to Australopithecines, are generally thought to have consumed high quantities of meat and, in the case of habilis, scavenged meat. Typically, early hominins are interpreted as having lived in polygynous societies, though this is highly speculative. Assuming habilis society was similar to that of modern savanna chimpanzees and baboons, groups may have numbered 70 to 85 members. This configuration would be advantageous with multiple males to defend against open savanna predators, such as big cats. Homo erectus had a more modern gait and body proportions, and was the first human species to have exhibited a flat face, prominent nose, and possibly sparse body hair coverage. Though brain size certainly exceeds that of ancestor species, capacity varied widely depending on the population. In earlier populations, brain development seems to cease early in childhood, suggesting that offspring were largely self-sufficient at birth, thus limiting cognitive development through life. Erectus was an apex predator. Sites generally show consumption of medium to large animals, such as bovines or elephants, and suggest the development of predatory behavior and coordinated hunting. Erectus is associated with the Acheulean stone tool industry, and is postulated to have been the earliest human ancestor capable of using fire, hunting and gathering in coordinated groups, caring for injured or sick group members, and possibly seafaring and art. Humans are the most abundant and widespread species of primate. They are a type of great ape that is characterized by bipedalism and exceptional cognitive skills due to a large and complex brain. Humans are highly social and tend to live in complex social structures composed of many cooperating and competing groups, from families and kinship networks to political states. As such, social interactions between humans have established a wide variety of values, social norms, languages, and rituals, each of which bolsters human society. The desire to understand and influence phenomena has motivated humanity's development of science, technology, philosophy, mythology, religion, and other conceptual frameworks. Genes and the environment influence human biological variation in visible characteristics, physiology, disease susceptibility, mental abilities, body size, and lifespan. Though humans vary in many traits, any two humans are at least 99% genetically similar. Humans are sexually dimorphic. Generally, males have greater body strength and females have a higher body fat percentage. At puberty, humans develop secondary sexual characteristics. Humans are omnivorous, capable of consuming a wide variety of plant and animal material, and have used fire and other forms of heat to prepare and cook food since the time of Homo erectus. Humans can survive for up to 8 weeks without food and 3 or 4 days without water. Humans are generally diurnal, sleeping on average 7 to 9 hours per day. Childbirth is dangerous, with a high risk of complications and death. Often, both the mother and the father provide care for their children, who are helpless at birth. Humans have a large, highly developed, and complex prefrontal cortex, the region of the brain associated with higher cognition. They are highly intelligent, capable of episodic memory, have flexible facial expressions, self-awareness, and a theory of mind. The human mind is capable of introspection, private thought, imagination, volition, and forming views on existence. This has allowed great technological advancements and complex tool development to be possible through complex reasoning and the transmission of knowledge to subsequent generations. Language, art, and trade are defining characteristics of humans. Long-distance trade routes might have led to cultural explosions and resource distribution that gave humans an advantage over other similar species.